to synthesize drugs that are of higher efficacy and of lower toxicity. So to achieve this, acetaminophen-based charcoal was synthesized using acetaminophen as the starting material, which was later reacted to the organometallic salt to give this particular compound, the adduct, which was on further demethylation using trimethylamine oxide, it gives the demethylated form. So these are the three compounds that were synthesized. The molecular docking was done using Pyrex, and metformin was used as a standard drug. Leishmanolicin was the protein docked with the compounds. And Biovia discovery was used for the protein preparation, while ADMET analysis was carried out using Swiss ADME and Protox 3.0 for the toxicity. The IR analysis of the synthesized compound as seen here. The first one is the acetaminophen-based charcoal, while the second one is the complex that was formed, while the third one is the demethylated form. And for the complex that was formed, the two carbonyl band was observed, which was not in the acetaminophen-based charcoal. And these are the coordinated carbonyl band, and there's a shift at the 450 band of this complex spectra, which indicates the ion to carbon coordination. And for the demethylated form, there's a disappearance of the band at 450, and also one of the carbonyl was seen to have disappeared too, which shows that the demethylation occur. And for the molecular docking that was performed, the synthesized compound is seen to have lesser binding affinity, which shows that they are than the standard drug, which shows that they have more stability and also interacted better within the receptor, within the active pocket of the receptor. And for the ADMET analysis, the boiled egg model shows that the acetaminophen-based charcoal and the demethylated form will pass the blood-brain barrier, while the molecule 2, which is the complex that was formed, they have good absorption ability.
And confirmation of formation of your compound.
Okay. Um, okay, I will note that. Although the FTIR will show the coordinated carbon and then the ion to carbon bond. So that was why we just chose to go for FTIR. But okay, I will note that. I think that will help the characterization better. Thank you.
Yeah, sure. So is this
Yeah.
your PhD work or meaning postdoctoral work?
Masters.
Master work. Okay, okay, great, great, very nice. So I
Thank
think
you.
Hasinul is want to ask some questions. You
Okay.
can unmute yourself. Yeah.
Yeah, are you able to hear me, Rachel? I have a questions to you. That means you have mentioned that you have used Pyrex software for molecular docking. Am I right?
Yes, I did.
Actually, I would like to know about a thing. I am also a presenter of this session, but I would like to ask you that means the result we got from Pyrex molecular docking, is it provides a, you can say, valid result or valid binding affinity or not?
The binding affinity, the Pyrex will reveal it. It will show the binding affinity because it includes the autodoc vena and also the autodoc.
We know that about you can say binding affinity is presented in negative below, and actually
Yes.
which was your, you can say, stable conformation. I would like to know about the, you can say, negative energy of molecular docking or uh, stable conformations. Would you like to share your you can say PowerPoint presentation again kindly, just for 30 seconds?
Share. Okay. Let me share. Okay. This is it.
This is it. And
Yeah.
what was your highest negative value for the molecular docking?
Okay. Yes. The highest negative value is for the one of the synthesized compound, which is demethylated. And the standard drug has the minus 5.0, which is higher than these other ones. So the one with minus 8.1 will bind better compared to this minus 5.0, followed by
Actually, have you compared this result with any standard drugs or standard compound?
standard compound is the metformin, and the standard compound is the one with this minus 5.0,
Okay.
which is the highest. Yes, which is the highest. So the ones with this lowest, starting from the minus 6.4, is for the acetaminophen based chacun, while this minus 7.4 is the organometallic compound.
Okay, okay.
The
I
minus
got it.
8.1 is the demethylated.
I got it.
So
Yeah.
it's binded better.
Okay. That means you are trying to say that your binding
Do
affinity
you
is
understand it?
greater than the standard compound. So it really, you can
Yes.
say, applicable for, you can say, clinical trial or something like this in the next procedure. So thank you, Rachel. Okay, I'm
Okay,
done with
Yeah.
you.
thank you.
Thank, thank you. I will
Okay.
take one question from Professor Priyani. Yeah.
Yeah.
Oh,
Can
okay.
I know that how you going to relate your studies to UNSCGs? How,
Okay.
you understand what I said? How are you going to
Mhm.
relate your studies to sustainable development goals?
Yes, for the Sustainable Development Goal 3, says that the well-being, good health and well-being, providing a drug that is of lesser toxicity will
Okay.
help a good health and well-being, and of course, also of higher efficacy.
Thank you. Thank you. That's all,
Thank you.
Sharma.
All right. Thank
Yeah.
you.
Thank you. And you have any question? I think
No.
then
So I had
because
typed my question in the chat, and so
Okay.
perhaps Rachel could answer when she gets a chance.
yes, that would be great. Yeah. So
Right.
now we can move to the next presenter. Rachel, can you unmute yourself? Mute yourself. Yeah.
Mhm.
Great. So second. Yeah. Second presenter is poster number P126. I think they have uh, slides or Fili- Filipina Pong. So from Department of Chemistry, Ghana, KNUST University. So their title of poster presentation is Quorum Sensing and Efflux Pump Inhibition by Phytochemical from Celestia debris. So do you want to present or shall I present?
And please, I would want you to share the screen for me.
Shall I share the screen?
Yes.
Okay, great. GCMS analysis of corn cob waste towards sustainable pharmaceutical use. So again, they haven't shared the slide. So Peter, you want to present here or shall I share the screen? Are you around?
Here.
Yes. So
I can see it.
and I'm asking because maybe somebody has joined from different.
Yeah, I know. That's
Yeah.
right.
So there. So I think we can again now move to the next presenter, post number P129, and Ilu Oluwaseun Akindele from University of Ibadan, Nigeria. And the title of the presentation is Identification of Potential Anti-Malaria Lead Compound from Selected Plants, Identified Phytochemicals, a Computer-Aided Drug Design CAT Study. So are you around?
Yeah,
Yes. So you want to share your slides or shall I present?
please do. Please
Okay.
present for me.
Okay. Is this your slide, right?
Yes, thank you.
Okay, go ahead.
Good day. My name is Akindele Oluwaseun. My work focuses on the identification of potential anti-malaria lead compounds from three selected plants phytochemicals using a computer-aided drug design approach. Malaria remains a serious global health problem, particularly in the sub-Saharan Africa and parts of Asia. One of the biggest challenges is the growing resistance of Plasmodium falciparum to existing drugs, which creates an urgent need for new, effective, and affordable treatments. Plants have historically been valuable sources of anti-malaria drugs, for example, quinine and artemisinin. And this motivated me to screen phytochemicals from three selected medicinal plants for their potential anti-malaria activity. The methods. In this study, 131 phytochemicals were retrieved from data bank, which was reported in the literature for the anti-malaria um, agent, ability. The target protein was also chosen, which was a Plasmodium falciparum lactate dehydrogenase, which is so crucial for parasites. Then the phytochemicals were retrieved from database, PubChem to be precise, and they were screened, they were optimized to know their best and stable conformal position. Thereafter, they were screened using ADMET and ADMET Lab 3.0 for their ADMET screening and analysis. The ones with positive outcome, the one that had good absorption, distribution, metabolism, excretion, and the good toxicity outcome were also tested for their drug likeness, using Lipinski rule of 5 as a standard. Then they were docked using orthodox vena against the target protein. The protein was also cleaned, viewed and cleaned, removal of water molecule and other interactions that could interfere with the docking um, docking process. So the result from the docking analysis, it was discovered that isolidine, one of the phytochemicals retrieved, had better binding affinity compared to the standard drug. The standard drug used was artemether, one of the drugs existing that has been used for the cure of malaria. So from the binding score, it was discovered that isolidine had minus 7.6 kilocal per mole compared to the standard drug that had minus 7.1 kilocal per mole. There were other compounds that had good had met and bioavailability results, but they had less binding affinity compared to the other one. And compare also the binding poses, which is being reflected in this place. The binding poses and their interaction are shown, and none of the compounds had hydrophobic interaction. So no hydrogen bonding between hydrophilic interaction. So none of them had interaction, hydrogen, oxygen and hydrogen interaction. So in that case, the standard drug and the best compound based on the binding affinity, they had the same binding interaction. They were bound to the active site. And also from this bioavailability radar, it is expected that anti-malaria drug doesn't pass the blood-brain barrier because it is not a central nervous system drug. So these drugs, isolidine, did not pass the blood-brain barrier. Also, the absorption, it had small absorption. They do not have all of the compounds actually had little absorption. So considering the radar too, this one that had this pink, um, the bioavailability radar is also explaining the flexibility, the polarity, and from this we are able to discover that the best out of these compounds is lumatilhol. It has the best about availability rather compared to other drugs. Now the conclusion of this is that okay, this bar chart is showing us the binding affinity of the compounds to the target protein. Now, the conclusion is that although isolidine had a very good binding affinity, but it has little, little bioavailability, and also the hardness result is not as good as lumatilhol. I considered lumatilhol because if it can be optimized, it is possible if it has a good binding affinity scoring function, which shows the effectiveness and efficacy of the compound to the target. So compared to the standard drug artemeter, it has no good bioavailability, it has no better binding affinity, it has no better absorption, distribution, metabolism, and even toxicity results. Thank you.
So now the session is open for discussion. If anyone have question, yes, Professor Priani.
Yeah. Now, when you do this, when you're looking at the bioactivity, did you compare with the standard drug?
Yes. Yes.
Yeah. So then how about the activity when compared to the standard drug?
The standard drug had minimal, in fact, the bioavailability radar is not as good as the compounds that were screened. Um, I mean, they were actually given here from my studies. These three compounds, isolidine, lumatilhol, and gamma cadhenin, had better bioavailability compared to the standard drug.
Okay, good. Okay.
Yeah, yeah. I think Hasanullah want to question. He raised his hand.
So may I ask the question?
Yes, yes, please.
Okay, my questions to the presenter. Actually, for optimization, which software have you used? Which kind of software have you used? For we know that about in case of molecular docking, at first we have to optimize the compound. So for optimization purpose, which kind of software have you used?
Pattern 14.
Yeah, could you repeat the answer?
Is pattern 14. I did conformal search and also optimization, optimized using DFT.
Okay, actually, for DFT or this kind of optimization, we need, you can say, Gaussian software or something like this. Have you used Gaussians?
I use pattern 14.
Okay, I got it. Thank you.
I think we can now move to the next presenter. And next, now the poster number we have P130. So Tomilola Ekingbade and from Federal University of Technology, Nigeria. And the title of the presentation is In Silico Design of Novel Ercocarpus Atlas Dried Compound Targeting the MYC Max Heterodimers. So, Tomilola, are you around here? Cannot see her name on the screen, but we can move to the next presenter. If later they join, then please type in the chat box so that we can share your screen later. The next presenter is post number 131. Adekemi Temitope Alade. Sorry if I am not pronouncing accurately. Again from Nigeria, Joseph Ayo Babalola University, Nigeria. And the title of the presentation is The Chemistry, Biological Activities, and Possible Industrial Application of Plumeria rubra, which rainbow flower essential oil. So, Adekemi, are you here? Not available right now.
It's a message. I saw the message. What is the
Sorry?
She said, Anika Brown. There's somebody else. So it's okay. Go ahead. No, she's not here.
Yes. So now we will move to the post number 132, Nitharshan Ananthalingam. So University of Colombo, Sri Lanka. And the title of presentation is Development of Dual Carrier CMC PEG LDH Nanocomposite Hydrogel for the Sustained Release of Micania Chordata Phytochemicals for Wound Healing Application. Nitharshan, are you?
Yes, I'm here.
Yes.
Yes.
Hello,
So
everyone.
shall I present or you want to present?
Either way is fine.
You can share
present,
your
sir.
screen. Okay. Just wait.
So,
Slides, right?
yes.
Yes. Okay. I'm sharing.
Thank you.
I think you can start the screen.
Yes, I can now.
Yes, you can start.
Good afternoon, good evening, everyone. My name is Nitharshan Ananthalingam, and I'm from University of Colombo. And this is my master's thesis research, and I connected. I tried to connect this with the UN sustainable methodologies. So chronic wounds are a serious clinical challenge because of the high infection risk, delay in healing, and frequent recurrence. And usually the traditional methods that we use fall short in providing long-term protection or controlled delivery of the therapeutic agents. So To address this, what we did is we explored a hydrogel nanocomposite system that can combine the structural support with the controlled release of the bioactive plant extract. So this plant extract that I used is Mecania cordata, a medicinal species that is known for antimicrobial and anti-inflammatory properties. And on the carrier side, I used a double carrier system or dual carrier system where the plant extract was incorporated or they encapsulated into a magnesium aluminum layered double hydroxide nano composite that was used as the primary carrier, and then the nanocomposite was embedded in a hydrogel that was synthesized using carboxymethyl cellulose and PEG. So this was used as a secondary reservoir. So initial control release was done by the primary carrier, and the secondary carrier actually supported the secondary release to further slow down the release and sustain the plant extract more time in the wound so that the wound connection time for the plant extract would be high. So pristine LDH was synthesized with under a nitrogen environment at 10 pH with a 2-to-1 molecular ratio of magnesium to aluminum. And then the hydrogel was, or rather, then the nanocomposite was prepared with the plant extract encapsulation using a DMS or solvent assistant ion exchange. And after preparing the nanocomposite with the plant encapsulation, we did a gelation into the hydrogel precursors, the precursors of CMC and PEG, where we used the citric acid as the cross-linker between these two. So citric acid using the diester bond, it cross-linked the carboxymethyl cellulose and polyethylene glycol, and within the structure itself, the nanocomposite was captured to kind of accommodate the protection of the plant extract as well as the slow release. So after the synthesis, we went into, first of all, the confirmation, characterization, as well as then the application studies where the biocompatibility as well as the cell migrations were tested. So that I would be discussing on the second slide. So on the release profile side, as you can see in the picture on the left-hand side, the nanocomposite LDH actually released about 95% of its components in 3 hours. However, when we combine that with the hydrogel, the release profile was 3 hours, only 70% was released. So it was elongated to at least 5 hours for the complete release. So we achieved a sustained release. And then we tested the biocompatibility using the MTT assays, and on the MTT assays side, over 90% of the cell viability was achieved in the Vero cell line that we used at the lower concentrations of the hydrogel as well as the plant extract. So both plant extract and the plant extract incorporated hydrogel showed over 90% of the cell viability on the Vero cell line. Although we did the cell monolayer test, it was not tested on a in vivo testing. And then combining that with the scratch assay, we went ahead and did the scratch assay for cell migration on the same Vero cell line. So on the Vero cell line, when we tested the assay, we used the negative standard. So the serum with 1% of 1% serum was used as a control, and hydrogel was tested on it. And the usual cell migration that was seen with the standard was in 24 hours, 22.78%, and hydrogel showed a 95.75% of cell migration in 24 hours, and in 48 hours, hydrogel actually has closed the complete scratch compared to only 46% on the standard, which showed us that the hydrogel composite that we have synthesized during this assay or during this project has significantly assisted the cell migration to keep the bioactives in the wounds long time, thus enhancing the wound healing abilities. And although we tested the encapsulation efficiency to understand and ensure that what sort of encapsulation has been occurred with the bioactives that we extracted from the plant extract, and the initial LDH has shown 81.36% of encapsulation, and when we did the second embedding into the hydrogel, that came down to 72.58. According to the according to the studies, 72.58 is a good level that we could consider for further development. And what we tested here is on a on a cell level and a monocell layer, and we wanted to on a further level, we wanted to actually test it on an in vivo testing using a mice cell or using a different animal mechanism to understand if this translates into a actually usable, realistic characteristic or outcome for us to actually can support a sustainable, low-cost wound healing gel that can be used to heal chronic wounds. Thank you.
Great, thank you. So I think before jumping to other, I want to ask you one question, dear. So because with the hydrogel, it is very challenging to check the site toxicity. So how you tried, meaning what kind of method you have used with the hydrogel, because they have property of having water, absorbing water. So and you tried the toxicity with whole LDH with hydrogel system or only the hydrogel?
So we tested actually all three. So we tested the plant extract itself first, and then we tested the plant extract incorporated LDH separately, and then we tested the hydrogel separately. Although I'm hearing a noise.
Yes. So yeah, continue.
So we actually had challenges with the hydrogel because of the mechanical stress that hydrogel introduces on the cell. Even though hydrogel may not have been as toxic as it would have seemed, because of the mechanical stress that it caused on the cell line, the biocompatibility that we or cell viability that we saw was about 92 to 95%, which actually could have been or may have been, little more than that if that mechanical stress wouldn't have been there. But we tested all three, plant extract alone, and then LDH incorporated plant extract, and then hydrogel after incorporating the LDH into it, all three.
Okay. So yeah, so I think Professor Priyani, please.
It's fine. There are several who wanted to ask questions, so let them ask now.
Yes. Okay. Okay. Great. So I think Arif want to ask question.
Yes, please. The presenter,
Yes.
Please tell me.
Hello.
How you determine the percent encapsulation of this hydrogel?
How did
Can
I
you explain the methodology and which technique you use to determine the percent encapsulation?
Encapsulation.
Yes, percent encapsulation efficiency.
So we tested the encapsulation efficiency for plant extract incorporated hydrogel, or the incorporated LDH first, and then the hydrogel second. So initially, plant extract was rather the LDH was destructed, and the plant extract was released into a tricyclial system, and then using the plant extract standard curve that we already prepared, we calculated or rather used a UV visible measurement, because we already had a standard curve based on the UV visible that we did for the plant extract, and then after the destruction of the LDH, whatever that is released on the tricyclial buffer system, we checked UV visible concentration for that as well, and we compared the concentration against the plant extract concentration to understand how much of the plant extract that we initially used in the start of the synthesis was translated into the final product.
Okay, thank you. Thank you so much.
You're welcome.
Thank you. I think Muhammad Kavish want to ask any question. So Muhammad Kavish, you want to ask question? You have raised your hand. I think so.
Can I ask a question?
Yes,
then?
yes, please.
Yeah.
Yes.
So now you said that you did anti-inflammatory assay. How did you do that?
Not the anti-inflammatory assay, ma'am. We did only the cell viability assay and the wound healing assay, scratch assay.
Okay. Wound healing assay, you did
Using the
scratch
scratch on the
test?
virocell line.
Is it a scratch test that you made a wound and you see the gap? Is that what you did?
Yes.
Okay.
So on the virocell line, we made a scratch and observed in 24 hours, 48 hours.
Yep.
To see the closing.
Okay. Good. Okay. Thank you.
Okay. Nice.
Thank you, ma'am.
So I think we can move to the next presenter.
Thank you, everyone.
Thank you. This is post number P133. So, yeah. So, Hafiza Aksha Arif and from University of Karachi, Pakistan. And the title of presentation is A Promising Strategy for Ocular Inflammation, Nanoscale Lipid Methyl Pre-Denissolone Conjugates for Targeted Retina Pigment Epithelmia Therapy. So, Arif, shall I present or you want to present from your side?
Please, you will present my slide.
I think you haven't, meaning we have your poster, but just wait for a second.
A stealthy, undeformable targeted delivery system, such as lipid methylprednisolone conjugates loaded nanotransposomes, to precisely put out the fire. They involve to enhance the drug stability, encapsulation efficiency, sustained controlled drug release, specific tissue targeting, high encapsulation efficiency, and high penetration and modulating the expression of genes associated with inflammation and pain within the RBE cells. Next, in the methodology, we have synthesized two novel methylprednisolone double ester conjugates that were conjugated by aliphatic chain by using the aliphatic acids, and to methylprednisolone by using double ester linkage like succinate or glutarate. These are linked as a spacer group, and we elucidate these our newly synthesized compounds using spectral techniques, either FTIR, proton NMR, carbon 13 NMR, HRMS. And then we formulated these newly synthesized lipid methylprednisolone conjugates into a nanoscale, such as targeted drug delivery system like nanotransposome, by using emulsion evaporation method. And then we formulate these nanoformulations physicochemically characterized using dynamic light scattering technique and some microscopic techniques like FM, SEM, and TEM, like transmission electron microscopy, and UPLC is used to determine their percent encapsulation of these nanoparticles. And the release study of these that were performed in murine plasma, and the cytotoxicity and the cellular uptake study was also performed. In vitro biological activities assessed using real-time PCR analysis by following cDNA synthesis and then RNA extraction from treated RBE cell lines. So we induce an inflammatory stage on our RP cell lines using PGN, peptidoglycan, that's a chemical that induces the inflammation. And our data from qPCR is a true measure of success. So in the result discussion, the vehicle is engineered to overcome the RP's defenses. It's small, as the DLS graph represent that their size is smaller, 100 to 140 nanometer. And these are the stable, we show the graph that representing the stability of these nanoparticles throughout over a month. And these are stable and highly loaded. And the percent encapsulation is high, greater than 90%, and has a negative charge. Its stability charge is this is a zeta potential that is a minus 24 to minus 30, and these has a stealth and penetration. So our physicochemical characterization confirms these are the ratified from the DLS, SEM, and TEM that's confirmed the uniform stable spherical particles. The cytotoxicity assay was confirmed that these are the completely safe for the RP cell lines up to a 50 micromolar. And in murine plasma, we study their release study. These nanoparticles don't jump down their cargo. They provide a prolonged, controlled, sustained release with nearly 70% of the drug delivered over 48 hours. This means reduced dosing frequency for the patient. So next we perform the cellular uptake studies by using, firstly we synthesize our nanoparticles by tagging with the rhodamine dye. And these are we visually confirmed their success. They were rapidly and successfully taken up by the RP cell lines, localizing precisely where they are needed inside the cell cytoplasm. So our nanolipid methylprednisolone didn't just suppress the fire, they are engineered the RP's defense mechanism. They significantly suppress and the most aggressive pro-inflammatory genes. The fire starters like TNF-alpha, we use different primers, pro-inflammatory primers, pain primers, as well as the anti-inflammatory primers. Pro-inflammatory primers like TNF-alpha, IL-6, IL-8, and IL-17, but the anti-inflammatory IL-10 and IL-4 and pain inflammatory marker, these are the 6-HCl2 and PDY-N. These results shown there concludingly our targeted delivery system are a highly promising strategy for targeted ocular therapy, and by delivering a potent anti-inflammatory agent precise to the RPE, so we can achieve better therapeutic outcomes with the potential for drastically reduced systemic side effects. So our stealthy targeted nanosubmarines hold the key to defending the retina's Kessler wall and preserving vision. Thank you so much.
Thank you, Hafiza, for the nice presentation. So now the session is open for the discussion. Yeah. So I want to ask initially, so Hafiza, what about the stability of this particle with this drug? And what was the PDI that you got initially with, I mean, when you freshly prepared this particle?
Freshly prepared particles have a PDI 0.1 to 0.13. But how we perform the stability, we check their size, PDI, and zeta potential till a month of these nanoparticles, and we store these nanoparticles at room temperature as well as 4 degrees Celsius. So these show the best stability over a month with the size with a little change just in a numbers like 110 to 112, 113 nanometer size, and PDI is with 0.12 to say 2 to 1.1 to 19 as, and zeta potential is also similar in change.
Okay, and also just want to know about that. You are doing your chemistry work also and the biological work, or it is when you are doing in collaboration this work?
No, I have done this overall studies by self.
Okay, very nice. Okay, Professor Priyani.
Yeah, so did you do this identification of compound using NMR you did by yourself?
Yes, I have performed by self NMR and also used topspin spec software to determine this elucidation as well.
So the 13 carbon of your compound, right? You your compound, I saw that you have a carbonyl, two type of carbonyl groups. One is the alpha beta unsaturated one. Other one is ester carbonyl. How did you identify these two?
Because of its chemical shift values, And
Yeah.
as most electronegative group is at the highest value as compared to the other one. So the
In
two carbonyl
your one, can I ask simply, so which one has the higher chemical shift?
double oxygen carbon is at the high position.
Okay, good.
Meeting
Good.
Thank you, ma'am.
Okay,
That's
and
all.
also
Excuse me, sir,
this
I can't understand.
yeah, so this UN Sustainable Development Goal, how you understand your work is going to benefit, meaning other than this SDG 3 number.
Yes, actually, this is relevant to we perform this inflammation disorders like ocular disorders in which eye-related dry eye disease, age macular degenerative disease. So we have to design them for to treatment of these disorders. So that's why I have choose the good health well-being portions.
Okay, okay, thank you.
Thank you, sir.
Thank you. So we can move to the next presenter, post number P134, Sarita Beg from Jinnah University of Women, Pakistan. And the title of presentation is Green Synthesis of Silver Copper Oxide Zinc Oxide Nanoparticle using Lactuca cereola extract and evaluation of their pharmacological activity. So, Sarita, are you around here? Yeah, I
Yes,
think.
sir, I'm
So,
here.
so you want to present or shall I share the screen?
Sure, I will share my slides here.
Yeah, go ahead.
It's
Yes,
visible to all of you.
yes. You can start now.
Hi everyone, I'm Sarita Beg and today I am presenting my bachelor's research work. Well, as we know that traditional nanoparticle synthesis methods are not eco-friendly and environmentally harmful. So by keeping this in mind, we have used biological synthesis method and we synthesize metallic nanoparticles and thenceforth we have evaluated their pharmacological activities. Well, the objective of this research is comparative analysis of silver, copper oxide, and zinc oxide nanoparticles and their evaluation of pharmacological activities. As you can see here in the diagram, first of all, we have taken Lactuca cereola leaf extract to made leaf extract in the water because this plant is a medicinal plant and contain a phytochemical constituents which helps in reduction of metal salts. Well, we mix both of the solution and kept on a stirrer and provided required conditions. And after completion of reaction time, we have observed color change. For silver nanoparticles, we have observed dark brown color. For copper oxide nanoparticles, we have observed dark black color. And for zinc oxide nanoparticles, we have observed yellow color. This is the indication of production of nanoparticles. As well as we also check the UV visible spectroscopy and check the absorbance of nanoparticles, and we got the expected results. Well, to check the particle size and zeta potential, we have performed DLS, dynamic light scattering technique, and we got expected result for silver nanoparticles. We got below 100 nanoparticles size, and for copper and zinc oxide nanoparticles, we got above 100 nanometer particle size. This is due to aggregation of phytochemical constituent with particles. Okay, so we have also checked the FTIR of our plant. Well, we have checked and we observed the different phytochemical constituents and different functional groups. As you can see here in the spectrum, we got a ether group, carboxylic group, and a carbonyl group, which helps in reduction of metal salt. Okay, so to check the topography and morphological structure of these nanoparticles as well as the size distribution, we have performed AFM. atomic force microscopy, and scanning electron microscopy. As you can see here in the images, the particle size are small, circular, and it's acceptable. And we have and we have compared our result with reported results. And the size distributions are also in the nanoscale range. Well, after this, we have checked the antibacterial and the antioxidant activities of these metallic nanoparticles. And with the help of the pH scavenging activity, we have checked antioxidant activity of these green synthesized nanoparticles. And all of the nanoparticles shown the antioxidant activity, and the silver nanoparticle shown the exceptional result. Likewise, we have conducted antibacterial analysis of these metallic nanoparticles against E. coli and S. aureus, and the positive control was moxifloxacin, and we have got a zone of inhibitions. And with all the nanoparticles shown the antibacterial activity, and again here silver nanoparticles shown the exceptional result. In conclusion, we have synthesized silver, copper oxide, and zinc oxide nanoparticles using Lectrica cereola leaf extract, and we have checked the antibacterial and antioxidant activities of these nanoparticles, with silver nanoparticles showing the enhanced efficacy. In future, we will focus on optimization at large-scale production, and as well as we will work on another applications of these metallic nanoparticles in a sustainable way. These are some references for your record. This research had been conducted at Jinnah University for Women, Karachi, and at ICCPS University of Karachi. Thank you for listening me carefully.
For the nice presentation. So now the session is open for discussion. You can stop sharing screen so that we can discuss this.
Sure.
So yeah, Sarita, so why you choose this meaning this plant leaf? So any specific reason of choosing this plant, and other what do you understand with the UN Sustainable Development Goal?
Well, the plant Lectrica cereola I have selected because this is a medicinal plant, and this plant have so many medical benefits for heart patients and as well as for diabetic patients. That's why by keeping in this mind, I have selected Lectrica cereola leaf extract. And the method which I have used in this research, I use biological method, and this is a sustainable. And again, the source which I have used is a renewable source.
Okay, no, I'm just want to ask that what is UN Sustainable Development Goal? What it stands for?
Of course, it stands for to use the biological activity and to protect our environment from harmful things.
Okay, thank you, Sarita. Professor Priyani, yeah.
So shall we give the chance to the others? There are two others.
Yes. So yeah. So Muhammad Kavish.
Hi, are you listening to me?
Yes.
Yeah, I just wanted to ask, did you perform any stability studies of these nanoparticles? Because you are preparing with white green synthesis, so you need to have some stability data as well with this.
Well, so far we haven't checked the stability of nanoparticles, but in the future we will.
Okay.
Due to lack of time, we haven't checked the stability.
Okay, thank you.
And Arif, do you want to ask
yes, sir.
any question?
Yep. Please, presenter, tell me, have you performed the cytotoxicity of these design nanoparticles?
No, only I have conducted the antibacterial and antioxidant activities.
So what's the IC50 of these nanoparticles?
What, can you repeat it, please, again?
What's IC50 values of these nanoparticles?
ICP?
IC50 value.
I'm not. No, I didn't check it.
Okay, thank you. It's okay.
Okay.
So in that case, how did you measure the antioxidant activity? If you didn't get the IC50? Yeah?
Yes, ma'am. I got the results of antioxidant activities, and the silver nanoparticles shown the enhanced activity overall.
Yeah, how did you measure it? I mean, quantitatively, how did you present it?
With the help of BPPH assay I have performed.
Yeah, I know that. How did you present your results? If you didn't calculate the IC50.
I have calculated.
So you said no to Arif.
I'm not getting what you are actually asking.
No, Arif asked, what is the IC50 value you got for this? You said you didn't calculate. That's why I asked. How did you present your results? Am I right, Arif? Yeah. Okay.
Okay. So anyone have any other question, or we can move to the last, I think, then we can again come back to those who haven't joined us previously. So I think it's post number 135, Harrison Banda from University of Zambia, and the title of presentation is Synthesis Anti-Cyclotosomal Structure Activity Relationship Profiling of N pyridine 3-aryl benzamide by in silico and in vitro approach. So, Harrison, are you here? Yes. So, shall I present or you want to present?
I'll present.
Yeah, please go ahead.
Yes, I'm sharing already. Are you able to see it on that end?
Yes, yes.
Hello?
Yes, please.
Okay.
Please, can you click on the present view so that we can see clearly? Yeah.
Are you able to see it?
Yeah, you can start.
Hello?
You can start. Yes, you can start.
Thank you very much. Thank you. So, my study looked at a neglected tropical disease called schistosomiasis, which is the second most fatal tropical disease after malaria. Currently, we have over 700 million people either living with it or at risk, and actually over 300,000 deaths. So, the main species that are relevant to this part of the world in my country are Schistosoma hematobium and Schistosoma mansoni. In fact, in my country, all the provinces are infested with schistosomiasis, with the capital city where I live having the highest, almost 50% of the endemicity. In terms of the statement of the problem, the current chemotherapy against schistosomiasis is mainly praziquantel all over the world, but which has proved to be ineffective against immature parasites and has also started developing resistance and tolerance. And so, even though it's considered cheap, it's because of the subsidies. Of late, my country has had no much of the supply of praziquantel. So in terms of the literature review, a number of studies are going on around the world to find an alternative to praziquantel. In my country, in my lab, we started with molecules we call N-phenyl benzamides by my area colleague called Masebe Kanyanta. N-phenyl benzamides were made and showed some good activity. Unfortunately, those molecules called N-phenyl benzamides, which were picked up from MMV 687807, were not favorable in terms of the cytotoxicity and also the solubility. The cytotoxicity was too high, and the solubility was also too low. So I picked up from there, and I changed a number of moieties around the area molecules to introduce 2 nitrogen atoms in an N-phenyl ring to make it a pyridazine ring, as you can see. So in terms of the methods, I started with the benzoic acid, and also reacted with a 3-amino using what we call EDCI mediated amide coupling.
Question. If anyone can you Vandap, can you please stop sharing screen so that we can discuss this?
Okay, thank you.
Yeah, please stop sharing. Yeah, that's great. So yeah, Arif, you want to ask any question? You have raised your hand.
Yes, sir. What's the significance of your
Okay,
compounds?
these compounds have
For which application
less they have less cytotoxicity and better solubility again, and also appreciable activity against the schistosoma mannsonii parasite.
Okay, thank you.
Priyani?
So, can you hear me?
Yes, I can hear
you can hear me, right? So then, in your study, so what is the ultimate goal of your study?
The ultimate goal of my study is to find an alternative to praziquantel that will be a novel drug, since praziquantel was already showing resistance and is not effective against juvenile worms. So as you saw, I actually did some studies on juvenile worms, which are ongoing. And so far, my study has proved that my molecules actually are less cytotoxic and more soluble than the ones that we started with at the beginning of a slightly different type of compounds called N-phenylbenzamides.
Good, good. That's all.
Yeah. So while doing in silico study and meaning before synthesizing these compounds, so what sort of literature you have done? So why you particularly focus in this such compounds?
Network is bad.
Yeah. So meaning while you started literature before synthesizing this compound, right? So what kind of property of these compound give you the confidence that okay, we want to try different structure activity relationship? Meaning, what functional group you identify that making the impact on the overall activity?
Okay. Thank you. We have what we call an N-aryl benzamide moiety. N-aryl benzamide moiety was proved by Medicines for Malaria Venture in an earlier study I referred to in a molecule called MMV687807. So our job was to increase the solubility and reduce the cytotoxicity of that molecule, which was a hit compound from MMV Medicines for Malaria Venture.
Okay, so you have done both the cytotoxicity study and synthesis part, or you have collaborated with somebody?
I've collaborated, but I also went ahead to do hands-on work just to make sure I am able, because I'll proceed with this for my PhD.
Okay, very nice, very. So Arif, you want to ask any question?
Already.
Okay, because your hand is raised, so that's why. So okay, so I think Professor Priyani, I think that's all the list that Commonwealth has provided. But I want to repeat the poster number that was missed, and if somebody is present and they want to present again, then it would be great. So poster number what P127.
Sir, actually, I would like to interfere. I have a query to you. Actually, yesterday, actually, I couldn't present my poster. The poster number was P104. That means P104. But when I
Okay.
emailed the organizing committee, they mailed me that actually I can present my poster at 15 GMT. 15 GMT.
Okay.
That means after 16 minutes, according to the actually, may I present my you can say thesis or work today or right now, or I should wait for the
So,
next session?
Professor Priani, shall we allow
It
him?
should
Yeah,
be,
I
it
think.
should be,
Yeah,
let,
yeah,
let
yeah.
him.
You can present. Yeah, you can start. Yeah.
Okay, thank you, sir. Sir, are you able to, sir? Are you able to see my screen?
See your slide and yeah, hear you.
Okay, thank you, sir. Thank you, everyone. And I'd like to present my work that I have done in my master's thesis. And the work was basically synthesis, antimicrobial evaluation, docking and dynamic investigation of novel sterile monoperoxide. That was my, uh, you can say, Commonwealth Chemistry Posters ID. At the same time, poster ID was P104. And right now, I'd like to present my overall tasks that I have done. Actually, our objectives was basically, at first, I'd like to summarize the overall things. At first, we have chosen some compounds, the compound that have some, you can say, antimicrobial properties. And we have chosen methyl alpha D manoperoxide. That was our starting material. And in our chemistry laboratory, we have chosen this compound actually by study different kinds of, you can say, previous articles or scientific articles. And we have seen that actually methyl alpha D manoperoxide that have awesome, you can say, antimicrobial properties. And if we change the free hydroxyl group from this compound, we will be able to get some exclusive antimicrobial properties of our synthesis products. And with the view to actually we have done lots of things. That means at first we have run, you can say, experimental tasks. That is our starting material. And uh, actually with the help of some, you can say, pyridine and acylating agent, we have completed reaction. And in this reaction, to make the faster, we have used actually a catalyst, that means DMAP, dimethyl aminopyridine, for making the reaction faster. And similarly, when we have completed the first reaction, we have seen that actually that is the, you can say, sixth number free hydroxyl group. When we have completed, you can say, sterilization, then six number free hydroxyl group is blocked by the sterile, you can say, acylating agent. And that is our first product. And from this product, we have further done acylation reaction using different kinds of acylating agent like octanoyl chloride. And the number three compound is acylating agent is octanoyl chloride, and number four is, you can say, decanoyl chloride, and number five compound is, you can say, dodecanoyl chloride, and that is a, you can say, hexadecanoyl chloride acylating agent. So we have we when we have changed the acylating agent, we have found different percentage of product. And the percentage was, you can say, 81%, 76%, 83%, and 66%. That means compound 5 was, you can say, huge amount. The percentage was really high. That was 83%. Then when we have completed, you can say, this acylation reaction, we have found, you can say, 2, 3, 4, 5, and 6 number products. And we have done some computational calculation. That means we have draw the synthesized product structure using, you can say, Gaussian software. And then we have we have using smiles, and we have run, you can say, pass studies. Pass means probability of activity spectra of substance. That means our synthesis products have, you can say, specific activity or not. That means it have any antibacterial, antifungal, or anticarcinogenic property or not. That we have done. So I would like to uh, show you that actually, when we have done pass analysis, that means probability of activity spectra of substance, then we have seen that our compound 3 that show the, you can say, highest antibacterial and antifungal properties. And at the same time, I would like to move in the next session, next, you can say, slide. That will be easier for understand, because my poster is, you can say, uh, compressed kind of. Then when we have done, you can say, pass analysis, we have found that compound 3, that compound 3, 4, 5, and 6, that shows the highest antibacterial properties, and that is, you can say, comparable to the tetracycline. Tetracycline is a, you can say, standard antibiotic, you can say. At the same time, if you'd like to say that PA and PI means probability to be active, PI means probability to be inactive. That means we are seeing that the probability to be active is higher in case of compound 3, 4, and 5, and 6, in comparison to the probability to be inactive. And at the same time, we have found that actually compound 3 that show the, you can say, similarly antifungal properties that we have done from past analysis. And at the we have compared with some standard drugs like FCZ, that is called the, you can say, fluconazole, ICZ, that is called the, you can say, itraconazole, NOIS, that is called the nystatin, and TTC, that indicates the, you can say, tetracycline. And the next one, when we have done good antibacterial and antifungal properties, we have done you can say, some experimental tasks on the basis of past studies. When we have get a good score from past studies, we have completed some experimental studies using microbiology department or microbiology lab from our, you can say, university. And we have seen that Bacillus cereus, that is a bacteria, Escherichia coli, that is another bacteria, Pseudomonas aeruginosa, and Klebsiella pneumoniae. These are the 4 bacteria that we have done, you can say, these studies for, you can say, to check it out, its antibacterial properties. And we used, you can say, Diggs diffusion method, and we have found, that means our standard compound tetracycline, that shows the 22.33 you can say inhibition zone, that was its 100 microgram deciliter in Diggs or per Diggs. And on the other hand, we are seeing that our compound 3, that show, you can say, good or comparable score, that is, you can say, comparable to the tetracycline. At the same time, it show the, you can say, good score in case of you can say Escherichia coli. At the same time, that is not the fully equivalent or you can say comparable to the, you can say, standard drug, but from our, you can say, experimental drugs, that we can assume that actually compound 3 that can show the good, you can say, antibacterial properties. And at the same time, we have done some antifungal properties using, you can say, this you can say, fungal strains like Fusarium equiseri, Aspergillus flavus, and Aspergillus niger, and penicillin. And then we have seen that our fluconazole compound, that is a, you can say, antifungal you can say, drug that is used in, you can say, our pharmaceutical industry in our country. And there is a good you can say, pharmaceutical drug, standard drug. But our compound 3 that show the 63.89 antifungal drug, that means score, inhibition score. So we can say that our compound, that is a good antifungal drug that can replace the fluconazole. Those are you can say available in the market, or you can say pharmaceutical industry. So we are assume that actually our compound 3 that can replace the position of fluconazole if it is go through the, you can say, in the clinical trials. That is our assumptions from the, you can say, computational studies, sorry, experimental studies. When we have get the, you can say, good antibacterial and antifungal you can say studies score or inhibition zones, then we have done some computational tasks that is called the, you can say, molecular docking. From the molecular docking, we are able to check it out. That means our antifungal protein, that means we know that about each and every antifungal protein that has, you can say, specific protein ID. That means we know that about 3LD6, that is a protein ID of, you can say, fluconazole or Candida albicans. That is a fungus. Actually, when we do molecular docking using our synthesized drug compound against.
Hasinullah, can you please summarize your talk?
Okay.
Sorry to interrupt you. Can you please summarize it? Yeah.
Okay. I'd like to see. Okay, we have done molecular docking and we have done molecular dynamic simulation to check it out. That means our compound that can inhibit the that means fungal protein or not. And we have see that actually our compound that has, you can say, good you can say binding affinity -7.1. Fluconazole that show the -9.3. Our compound that show the, you can say, -7.9. Our experimental study and computational study that you can say corroboratively, you can say that actually this compound 3 that can take place the position of the fluconazole in you can say medicinal sector, or you can say pharmaceutical sector. That is our outcomes. And in these specific studies, we have done, that means we have synthesized our compound. At the same time, we have checked out the biological activity. That means it is antibacterially active or antifungal active. We have seen that actually our compound 3 that show that you can say more antifungal activity. And at the same time, we have checked out the computational, you can say toxicity determination using Protox 3 server. And we have seen that our compound that has, you can say, less toxicity. That means that at first, I mean, I have shown that actually fluconazole that has, you can say, some side effects and toxicity and hepatotoxicity, but our compound 3 that show that you can say lowest toxicity, we have confirmed from our, you can say, computational study using Protox 3. So that's all about my presentation. So if you have any question, then you can ask me.
Visitor, you can stop sharing your screen.
Okay, thank you, sir.
So yeah, so session is only, I think, one question we can take, and then I think there's one more in the chat box. I think if anyone have questions, they can ask the presenter in the chat box. We can go with the, I think, the last presenter. Or again, I want to ask if anyone, those who are in the session and missed the presentation, post number 127, post number 128, and post number 130, and post number 131. Is anyone here? Want to ask question, Hasanul Babu? So yeah, Professor Priani. I think there is one more in the chat box. They have asked that they also want to present today. So
yeah, it's me. It's Kavish. Yeah.
Yeah, Kavish. So Kavish, can you please quickly, present your, poster?
Yeah. Just a moment.
Please go ahead.
Sir, actually, I'd like to say something actually. Actually, my question is, when I have submitted my, you can say, abstract to, you can say, Commonwealth Chemistry Posters 2025, then I had shared my recorded video. That means we know that about in case of Commonwealth Chemistry Posters, actually they will evaluate the posters. That means my today's presentation at the same time recorded video will be part of the, you can say, evaluation.
So that we need to ask the organizer, or they might, because a different group has different chair and the judges, and so they, if you have submitted your recorded video, then that definitely they have screen in you, or you can ask them the organizer, and we are happy to present our remarks on your presentation.
Okay, thank you, sir. Thank you.
Okay, thank you, Babu.
Okay,
Yeah, please, Muhammad Kavish, go ahead.
So are you seeing my presentation?
Yes, we can see.
Okay, so the topic of my poster is highly functionalized pH trigger supramolecular nanowheel for target cancer chemotherapy. My work is specifically on liver and lung cancer because they are the fourth and sixth most abundant cancer across the globe. And among the possible therapies, chemotherapy is one of the most practiced option these days. But the chemotherapeutic drugs have poor bioavailability and have severe side effects, as well as they have poor target specificity as well. So over the years, various drug delivery system has been designed to mitigate these shortcomings. Among them, the stimuli responsive systems are the most preferred option because they used to deliver drug before when reaching their target. My preferred system are the pH responsive system because they releases the drug within the cell and endosomal and lysosomal pH. So my study is based on designing a system using a mesoporous silica nanoparticles and as a pH responsive stalk, I use the benzimidazole. So first I modified the pH responsive stalk on the surface of the nanoparticles, and then I use a modular quercetin, which is and then I modified these nanowave and I capped this nanoparticle with beta cyclotaxin to prevent the drug leakage. And my objective is to release the drug within the cell at endosomal and lysosomal pH. So in the methodology, I designed the first mesoporous silica nanoparticles, and then to I load quercetin onto the porous nanoparticle, and then I capped it with beta cyclotaxin. Because this is the new moiety, I also managed to establish